Hauke. So our speaker today is Alžbeta Chorvatová from Laser Center in Bratislava, University Bratislava, whatever this is. I'll get the details. Okay, International Department of Biophotonics at the International Laser Center in Bratislava. And Alžbeta is also a full professor and the head of Department of Biophysics at the University of Trnava in Slovakia. So this is, Alžbeta, this is even new for me. I heard a lot of Trnava, but I didn't know that you have so close relationship to them. So many of us probably know Alžbeta already. Alžbeta, for a long time, Alžbeta worked in Canada, in Montreal, and did a lot of work on metabolic film and metabolic spectroscopy in live cells, not only in algae and moss as today. She did investigations in live cardiomyocytes, which was very tricky to say the least. And now she is interested in evaluation of changes in mainly of natural endogenous fluorophores using spectral imaging and film. And she uses this to monitor non-invasively the possible pathological changes in animals and humans and human cells. And she is also interested as in the capability of algae or moss to react to environmental pollution and other challenges. So not quite surprisingly, the presentation of Alžbeta is investigating environmental pollution by means of spectrally and time-resolved imaging of natural endogenous fluorescence in algae and moss. So Alžbeta, we are very excited to see your presentation, and feel free to start.
Thank you very much, Wolfgang. And I would like to thank Becker and Hickl for giving me this opportunity to actually speak to you today. And what I would like to talk to you in this seminar and in this webinar is in fact pandemics that is a little bit different from that we are now dealing with, the COVID pandemics, but it's a pandemics of environmental pollution. In fact, we are having a lot of problems with the environment in these last decades. And this is related to many different phenomena. Those phenomena include the acid rain, heavy metal pollution, but also in the last decades, microplastics. When we talk about acid rain, in fact, this is a result of our everyday work, everyday transport, but also the plants that results in rain that is much more acid than normal water. You may know that oceanic water has a pH around 8, pure water is around 7, and normal rain is around 5 to 6, but acid rain can go down to 3, which is very close to battery acid and can have a very harmful repercussions on organisms. The next significant pollution is a pollution of heavy metals. This is resulting, of course, from the mining and industrial pollution, but also more and more agricultural and urban developments. So these heavy metals accumulating in seas and also in water can present a lot of problems. And finally, in the last decades, existence of a huge number of microplastics, of the plastics in the environment turning to microplastics results in food chain, so in animals and plants that are more and more accumulating these microplastics that we then eat and consequently can have problem with it. So these were the problems we wanted to focus at, and we believe that really the 21st century challenge would be to understand and prevent this environmental damage and to find appropriate scientific approaches to study its effects. So our solution to this problem was to test the use of advanced biophotonic methods to study the responsiveness of algae and moss to environmental pollution by means of changing of their natural endogenous fluorescence, of fluorescence that you have without any additional probes, and also search for remediation approaches. So what we chosen to use for the study was algae and moss. Why? Algae, you may often know, as something that is not very pleasant in the environment. But in fact, algae belong to a very large and diverse group of photosynthetic organisms. They can range from unicellular algae, like Chlorella or Dunaliella, I will talk to you today, to brown algae that can have 50 meters of length. And they play a significant role in aquatic ecology, providing food base for most marine food chain. In fact, we have a lot of advantages from algae. They are used for production of biopolymers, such as agar or alginates, but also energy sources, such as pure biomass. They're, of course, fertilizers. And in many parts of the world, they are also nutrition. We often use vitamins and supplements related to algae, such as spirulina in Chlorella or vitamin C and beta carotenes in Dunaliella. And of course, we can also use them as pigments, coloring agents, and stabilizing substances. However, more and more, we are realizing that algae are also very sensitive to the environment and to pollution. So they can provide sort of pollution controllers and monitor the pollution around us. That's why we have chosen them. And as an example of different system, we also choose a multicellular system of Bryophyta Moss, Fontinalis antipyretica, which has been basically used for decades to monitor air quality. And as they don't have the root system, they are really very sensitive to their environment. And there are studies showing them as sensitive biomonitors of pollution by expression, altered expression of antioxidative enzymes. So we have chosen these two approaches, these two types of samples, and our first aim was to examine their endogenous fluorescence by both the spectrally and time-resolved microscopy methods. So concerning the endogenous fluorescence for plants, algae, and moss, the most important part of this fluorescence is derived from chlorophylls. You may know chlorophylls very well because they are actually the photoactivable pigments that are the best known and the best capable of taking the energy from the sun. In fact, chlorophylls has this porphyrin-like ring structure with magnesium in it, and they are derived from protoporphyrin 9. Those of you working with cancer, you may have heard of this molecule very well, because it's actually the very important molecule for cancer detection. But in fact, when you use light and NADPH-dependent processes, you get chlorophylls, which in chloroplasts enter in photosystem 2 and are able to generate energy that is absolutely crucial for us, humans, and all animals on Earth, because thanks to that, we have oxygen, we have glucose, we have everything what we need for eating, and basically we can get the energy to work and to exist. So chlorophylls are fascinating because they actually take practically all the wavelengths, the spectrum that comes to Earth from a sun. They are particularly keen of the blue light and the red light. They are not that much interested in the green one. That's why they appear in green. And they emit a red fluorescence, which has the maximum around 680 nanometers. So we can quite nicely detect them. So what is interesting with algae is that when you have native algae in a good health, they actually appear green in transmission and they appear red in fluorescence, thanks to this chlorophyll fluorescence. However, if they are in stressful conditions and have some problems, they start to lose color and become transparent in transmission, and very interestingly, they turn green in fluorescence. So thanks to that, we can relatively sensitively look at responsiveness of these algae to stressors. So concerning the fluorescence, as I already mentioned, the most important fluorescence in these systems are chlorophylls, particularly chlorophyll A, that have fluorescence lifetimes relatively short, around 0.2 to 0.5 nanoseconds. They can go up to 3 nanoseconds, but it's most in the in vitro conditions. But in addition to the system, we also have in the photosystem many other pigments and also NADPH related mechanisms and NADPH related flavins and flavonoids. So we can also get the classical type of endogenous fluorescence that we can see in animals derived from NADPH and flavins that also have very sensitive lifetimes, particularly to metabolic changes. So we have very many opportunities how to actually study things going on in the algae by a natural way. So as a sample, we have actually chosen 3 different systems. First was the Chlorella algae, which is actually a sweetwater green algae. You can find it as pills in pharmacies. It's like spirulina. Basically, this algae was gathered from our university at Trnava. It's a collection of green algae, and they are basically they were isolated from the main drinking water supply. So concerning their excitation and emission properties, we could show that, as expected, the main excitation absorption spectrum was at the UV range up to around 500 nanometers, and then they have a second peak in the red spectral region. And concerning emission, the main emission was really in the red spectral region at 680 nanometers, but we also have NADPH dependent fluorescence at UV, and between 5 and 560 nanometers, we had also a flavonoid related fluorescence. The next sample that we have used was a sample of a marine phytoplankton, Dunaliella tertiolecta, which is actually a small unicellular algae of about 10 microns length. It has flagella, so it's a flagellate, so it has a very pronounced motility. It moves very fast. And what is important, it's covered only with a elastic plasma membrane, so we expected its high sensitivity to changes in the environment. Not to confuse this algae with the Dunaliella salina, which you may know from these red seas that sometimes happen where this Dunaliella accumulate a lot of beta-carotene and food starvation. Our Dunaliella tertiolecta was isolated from Mediterranean Sea, and it was in collaboration with Rudo Bošković Institute in Zagreb, Croatia, and was thanks to our Visegrad Fund. In fact, this algae had excitation and emission properties that were very comparable to Chlorella. What is important to mention when we talk about this algae, as I said, they are flagellates, so they are moving very fast. So we have some difficulties to actually find a way to immobilize them to have a good images. So we try different ways of immobilization, such as, for example, the hydrogels, but this was not very successful because in this case, algae were in 3 dimensions, and taking 3D images is really not straightforward. But thanks to our collaborators from Zagreb, we were able to use the polyethylamidine, which allowed us really to immobilize the algae so that we can also do long-term experiments. And finally, we have used the Fontinalis antipyretica moss, which was gathered commercially from the aquashop because it's actually the algae that you normally use in your aquarium if you want to clean the water in it. And it was done under regular light conditions. So concerning the pigments that we have found in the samples, and particularly in algae, we were mainly interested to prove that we have the chlorophyll A, which indeed is the case, but we also found the chlorophyll B, luteins, phaeophytins, and carotenoids, which were also proved by micro Raman measurements that has the expected peaks for carotenoids. So concerning the methodology, we have chosen to use a multimodal imaging. So to couple both the spectrally resolved imaging and time resolved imaging in order to gather the most information about the molecules and its environment, because for that the film is very important, that it really gives you information about the environment of the molecule. Precisely for spectral resolved imaging, we have used confocal microscopy by LSM 510 meter from Zeiss with excitation of 450 or 488 nanometers. And for film, we have used time correlated single photon counting by Becker and Hickl with BDL 473 nanometer laser and long pass 500. So with this setup, we were able to gather spectral resolved images in order to prove that really the most important fluorescence in our sample is the chlorophyll fluorescence. And we showed it for the excitation of 635 nanometers as well as for the 450 and 488 nanometers. So indeed, maximum emission at 680 was recorded in all of these cases. Now you may ask, okay, but where is our green fluorescence? And this was actually an issue because green fluorescence in comparison to this red one was really very small. So we needed to use two different detection systems in order to gather really information on the green fluorescence between 5 and 550 and the red fluorescence, red chlorophyll fluorescence. So in fact, we have created two recording windows, one between 5 and 540, and the second one between 600 and 680, in order to gather most of the information on the change of this fluorescence. So while we have used two different windows for the spectral components, for the lifetime components, we have used a single detector, and these components were isolated by advanced data analysis. In fact, as I mentioned previously, when you have native algae, the most important fluorescence is the red, so we really found a very strong intensity. And in this case, fluorescence lifetimes were rather short, between, it was 200 and 600 picoseconds. However, in the presence of environmental stress, most of the time the fluorescence intensity decreased, and fluorescence lifetimes were either shortened or we have a prolongation of the fluorescence lifetime, mostly related to appearance of this green fluorescence. So in order to analyze the fluorescence lifetimes, I mentioned that we have used the long pass filter of 500. But we have verified with a band pass filter of 650 and 750 nanometers that indeed the most of the fluorescence of the lifetimes that we can detect in our sample are derived really from the chlorophylls. The green fluorescence can only be seen when these chlorophylls are significantly attenuated. So when we considered how to analyze the fluorescence lifetimes, this is always quite tricky to choose how many components do you have in your system. We have opted for two components, but we verified that, of course, the system could be also analyzed with three components. But when we look at different changes in specific environmental conditions, many times three components were just not appropriate for this type of analysis. So finally, we rather opted for two components. And in addition to the SPC image, we also used a custom-made lifetime analysis, and I will show you a little bit later why. So our specific aim was, once we have this fluorescence characterized, to monitor how this fluorescence will behave and how the cell will behave under influence of different stressors. And we have chosen particularly the pH, heavy metals, light exposure, and also microplastics. So concerning the effect of pH in the chlorellas, we have basically worked very straightforward in a very straightforward way. We have adjusted pH to different solutions using acid citric or sodium bicarbonate, and we left the algae for up to an hour in the solution and recorded it at room temperature. Sometimes we have also used specific environmental modulators that are changing pH. And what we have observed when we looked at the effect of pH and fluorescence, we were quite surprised to see a linear decrease in this fluorescence with acidification. However, green fluorescence was practically untouched. Only at a very low pH, we started to see a rise in the green fluorescence. And we could show that this was really related to change in the chlorophylls, because it was really the spectra at 680 nanometers that were lowered. And interestingly, at a very low pH, we even see a shift in the fluorescence spectra towards red spectral region, most likely related to shutting down of the photosystem 2 and possibly other systems taking place. So, of course, we were interested whether this is somehow related to change in the environment of these molecules, and that's why we have used the fluorescence lifetimes. And interestingly, we have found that acidification resulted in shortening of the fluorescence lifetimes, but in very severe acidification, this was reversed by its prolongation. So indeed, when we looked at the TOR1 component, so the shorter component, we could see that acidification in a linear way induced the even further shortening of this component. However, the TOR2 was practically unchanged, but in a very severe acidification, it was prolonged, and this probably also contaminated our estimation of the TOR1. So from this result, we could see very nice correlation between decrease in the red fluorescence, a shortening of the fluorescence lifetime, suggesting that this decrease is really related to change in the molecular environment, not the number of the molecules. So next, we wanted to look at the effect of pH in Dunaliella algae. And as this was an international study, we have done the study in at the same time by looking at the changes of the heavy metal cadmium. In fact, while the pH was adjusted with citric acid, as in the case of Chlorella, cadmium was actually exposed to cells for 3 weeks, and it was added in a concentration of 1000 milligram per liter, which corresponded to around 600 microgram per liter of bioavailable cadmium. And this is around 200 times the concentration of cadmium in surface waters to really, in the laboratory conditions, induce a maximum effect. And what we observed was really something we didn't expect. In fact, Dunaliella did not react too much with change in the chlorophyll fluorescence. Actually, it showed to be very resistant to this kind of changes, but we have recorded this change in the rise in the green fluorescence, but only in the flagella region of the algae for both the cadmium and the acid citric. So here we really have an issue, because now we needed to find a way how to analyze those data separately in this very small algae that has about 10 to 50 microns diameter, separately in the flagella and in the body region. So how can we do it? We have decided to do it in such a way that actually the confocal images and the lifetime images were done at the same places of the image. And therefore, we could scan the LSM image, identify the presence of the flagella root and the flagella region, and then use this information for analysis of the film image. So what we have actually done is we have created a pixel mask that was based on this multi-channel information. The area around flagella was about 5 micrometer perimeter, and the next part was considered as a body, a region which allowed us to create this kind of mask. And from this mask, we could calculate the intensity of the body and flagella region separately. And we can use the same mask to project it to the film image and also calculate the fluorescence decays in these two regions separately. And when we have done that, we have observed very interesting results. While in the red fluorescence in the body region, this fluorescence was higher than in the flagella region, there was no change with cadmium or acidification at all. And in the green fluorescence, again, in the body, we didn't find any significant change, but in flagella, we find significant rise in the green fluorescence with both the cadmium and acidification. So next, we wanted to see what's going to happen with the fluorescence lifetimes in these regions. And again, we were surprised because while in body region, fluorescence lifetime did not change at all, and in the flagella region, cadmium didn't affect significantly the fluorescence lifetimes. Acidification produced a significant prolongation of the TOR2 of the fluorescence. So basically, we were able to distinguish, thanks to these film measurements, that there are two different processes going on. While in the case of cadmium, there is an increase in the fluorescence, this is most likely related to the number of the molecules that is increasing. While in the case of acidification, the increase in the fluorescence is rather related to change in the molecular environment. So this was quite fascinating. And actually, when we have done comparable experiments in the Fontinalis antipyretica moss, we were also able to show that in this case, the green fluorescence was increased and raised and led to prolongation of the fluorescence lifetimes. So our next question was to see whether this rise in the green fluorescence is something like a protective mechanism, something natural, or is it something that only happened in a specific conditions? So we wanted to test it with a light exposure. A light exposure is something that is also, the organisms are also affected by changing the light exposure in the nature. Our experiments, we decided to use laser-induced photo bleaching for this experiment. So in fact, under normal conditions, all the experimental settings that I have showed to you till now, we have done a lot of effort to keep laser illumination at a very low level, in such a way that it actually is lower than the solar irradiation during daylight, so the natural light that the algae get. But in these experiments, we wanted to stress the algae. So we wanted to stress them by laser-induced photo bleaching. So we turned the laser power on at 100% in order to really have a very strong effect and look at consecutive scanning. And the result in Chlorella spirulis was quite interesting. We observed very fast bleaching of the red fluorescence, and on the other hand, a rise in green fluorescence that, although you may not see it here because the difference between these two fluorescences is very high between the green and red, but it actually presented quite a long fluorescence lifetimes. In Dunaliella, we had again unexpected effect in regard to the fact that fluorescence of the chlorophylls bleached much longer than in the case of Chlorella. It took much long time, so it really showed that Dunaliella is able to cope much better with the stress. And when it started to change, this green fluorescence started to appear in the flagella region first, before the whole body responded. And in this case, again, the photo-bleached area had much longer lifetimes than in control conditions. In fact, we could show that this is actually transient effect. So while the bleaching of the red fluorescence is linear, the green fluorescence starts to rise, and this effect is transient first in the flagella region and then, much more importantly, in the body region. And these effects can be affected by stressors such as cadmium or acidification. So this really proved us that the effect that we saw was a real effect. It was a real mechanism that these algae use to somehow cope with the stressful conditions. So as we have done this first study with the Dunaliella and the exposure to cadmium, we wanted to go a little bit more into details concerning this effect of heavy metals. And in order to study the effect of heavy metals, we have decided to create our heavy metals and specifically heavy metal nanoparticles by femtosecond laser ablation. In this case, we have actually used a laser microworkstation, which has the spirit laser, and this laser has 1038 nanometers and femtosecond, 400 femtosecond pulse. It's normally used for 3D photopolymerization, but we have used it for ablation of metal discs in order to get nanoparticles of specific sizes, submicron sizes. And in fact, we have used different nanoparticles such as cobalt, zinc, nickel, and copper. So what we observed when we exposed nanoparticles to chlorella, we could see that there was a decrease in the green as well as in the red fluorescence for most of the heavy metals. Nickel, for example, doesn't have any effect, which was surprising to us, but it's probably because of the time of exposure, which was about 2 weeks. However, copper and zinc were able to decrease the fluorescence, but you can see also that even the number of these algae decrease. But this fluorescence is recorded from individual algae, so this cannot be due to the number of algae decreasing. So it's really showing some toxic effects. And these effects are really related to decrease in the fluorescence with the maximum at 680 nanometers, so with the chlorophyll fluorescence. However, fluorescence lifetimes, as in the case of the cadmium exposure to Dunaliella, did not significantly change, which is quite interesting, and it's really showing that in this case, it seems that heavy metals have mainly effect on the number of the fluorescence, chlorophyll fluorescence molecules than the environment in these algae. So in order to be able to compare this situation in a unicellular algae to a multicellular organism, we also look at the effect of nanoparticles in the moss. And here the situation was a little bit different, because now the nanoparticles were engulfed by the moss, and therefore they stayed there for quite a long time. And therefore they induced much stronger effects. On the one hand, you can see here the body of these cells is mainly related to chloroplasts, fluorescence of chlorophylls, and the green fluorescence is mainly in the intercellular space. So this fluorescence was increased with the zinc, but completely abolished with other nanoparticles, and copper even decreased significantly the red fluorescence. So we can see it here calculated. You can see even the blue fluorescence, which is actually sometimes some nanoparticles can be seen or as a metallic nanoparticles in blue. Here, what I want to show is really this rise in the green fluorescence with zinc and decrease with the others. And for the chlorophyll fluorescence, we have a rise with zinc and decrease with copper. The other didn't show significant effect. So when we then look at the fluorescence lifetimes, this time the situation was different from the algae. In fact, you can see very nicely that copper shortened significantly the fluorescence lifetimes, and we can really see it here where we have this very short lifetime appearing, and zinc provokes rather a prolongation of the fluorescence lifetime. So this time, as a difference to the algae, really this change in the fluorescence can be related also to the effect on the molecular environment in these cases. So in the last part of my webinar, I would like to talk to you about the effect of microplastics. Why microplastics? Well, really, in the last decades, it is proving that the presence of this large amount of microplastics in the wildlife is resulting in many of the food that we are eating is actually contaminated. And this contamination for a long time was considered as unharmful to man. So even now we consider that eating plastic is not dangerous. Well, it is not if you eat a little bit of it. But if you have a lot of dust from microplastics that comes into the food chain, now we have starting to have problems, medical problems related to its accumulation in both respiratory, digestive organs, as well as reproductive organs. So in a healthy person, probably having some amount of microplastics is not a problem. But when there is an additional disease, such as cancer, the oxidative stress provoked by this dust overload can hamper significantly, for example, the possibility of this person to recover from the disease. And there are even studies questioning whether even some problems of people with the COVID cannot be related to some amount of microplastics in their lungs. So it is becoming more and more an issue, and we are very pleased to be part of the international study that is now launched from the last December with the LASERLAB-EUROPE V project. It's one of the tasks is really to understand the effect of microplastics on the organisms, and also with the effort to find a way to clean waters and clean the environment from this pollution. There are more and more microalgal-based biopolymers for nano and microplastic removal that are proposed. And we are actually asking ourselves whether we couldn't use the whole algae or moss to do this job. So not to introduce any additional pollution, but really to use a natural environment to clean itself. So we are really only at the beginning. These are very preliminary studies that I will show you today, but our first effort is to test the commercially available microplastics. It's actually latex beads that are around 30 nanometers, and we could see capacity of algae to attach those microplastics. For the moment, after 3-day exposure, we didn't record any significant change in the fluorescence. However, in the case of Fontinalis antipyretica, we see a really nice capacity to engulf these microplastics. And as a result, we see rise in the green fluorescence and also decrease in the red one, related to a significant decrease in the fluorescence lifetime. So we see a very comparable effect that even the heavy metals were able to induce. So again, here we possibly have some ways where to go for our future directions. So in conclusion, I hope that I have demonstrated to you that algae and moss, endogenous fluorescence is a very precious tool that can be used and that can be monitored by both the spectrally and time resolve techniques. And then we can use it to study the responsiveness to environmental pollutants and therefore use it for ecotoxic risk assessment. And hopefully these natural organisms can serve us as sensitive biosensors of the environment, and in the future also use it for remediation, for removal of these pollutants. And as you could see, time resolve techniques helped us a lot to understand the real nature behind these effects. So we very much hope that by our work, we would be able to reverse this environmental pollution towards back the environmental beauty that we are all fascinated with. And we believe that this is the world we want to live in. And the work I have presented to you, you can find several of these articles online. There's an open access, so you can have a look at this work more in details. We are also preparing some LASERLAB webinars in spring, so hopefully if you are interested, check their website. That would be mainly concerning the data analysis. And finally, I would like to thank again Becker and Hickl for giving me this opportunity to speak to you today. And particularly, I would like to thank International Laser Center in Bratislava and my colleagues that helped me with this work, and also students and young investigators from the faculty of University of Silver Methodius in Trnava, Ruda Bošković Institute in Zagreb, particularly Nadica Ivušević Denardis and her team. And of course, I would like to thank local and also European funding, and of course, you for your attention. And I'm keen for questions. Thank you.